Azolla. Azolla is a water fern just like Salvinia, Marsilia, Pinnularia, Regnalidium. Hello friends, I am Dr. Antika Das, and in this video I am going to discuss about the reproduction and life cycle in Azolla species. Earlier, in an introductory video, I have discussed about the external morphology, anatomy, the fossil form, ecological as well as economic importance of Azolla and also its distribution. So this is the second part of that video, and a link to that video I have put in the description box. So in this part of this video, I will be discussing only reproduction and life cycle. Reproduction in Azolla, it is two types. One is the asexual reproduction, another is the sexual reproduction. The asexual reproduction in Azolla, it occurs by fragmentation of the rhizome, the branches or the fronds, even the fronds of the Azolla plant. So this method of fragmentation, reproduction by fragmentation, is pretty common, and sexual reproduction, it is only limited during the summer time. Sexual reproduction occur by spores, where the spores they germinate to produce the gametophytes, and this gametophyte in turn produces the gamete that fertilize to produce again the sporophytic generation. But sexual reproduction is limited to the summer season. And this Azolla is a heterosporous type of fern. Heterospory means that there will be two morphologies of spores available based on size and shape. The male spore is the microspore, or much smaller in size, give rise to the a small spore that germinate to produce the antheridium, the gametophyte that produces the antheridium. And this macrospore is a female, it is a female spore which produces the female gametophyte that in turn germinate to produce archegonium containing the egg cell. So this male is the microspore, and the female spore is the macrospore. So the spores, generally the spores are produced in sporangia on the lower surface of the leaf, but here, as because Azolla is a water fern, it produces the spore inside specialized structure called the sporocarp. This type of sporocarp we can also see in Marsilia as well as Salvinia. So this are the sporocarp is a specialized form of the sporangium, as because it is in a aquatic environment. All the aquatic ferns, they have the spores produced inside the sporocarp, and these sporocarps are related, they're positioned on the axils of the older leaf, So the fertile, when the plant turned fertile, so the leaves, the lower lobe, we know that the lobe, the Azolla has two lobed leaves. The upper lobe contains the anabena, and the lower lobe contains the reproduction is limited to the lower lobe. So this one, the lower lobe. So this lower lobe bears the sporocarp, and the upper lobe have a protective function. It protects the lower lobe sporocarp from excess desiccation or any other mechanical injury. So the lower lobe is reproductive in function and the upper lobe is protective in function. Here in this diagram we see that this yellow colored mass, this here, they are the microspore attached with massula, and this red colored structure is the microsporangium. And in the next slide I will show you the macrosporangium also. In this diagram we see that this structure here, in this structure, this one, it is the macrosporangium or the female sporangium, and this much larger structure here, let me draw it in another color. So this one and this one, this larger structure, it is the microsporangium, that is the male sporangium. So we see that the size of the male sporangium is much larger than the size of the megasporangium, that is the female sporangium, which I have pointed in red. So just because the female sporangium, the female spore, megaspore, is much larger than the microspore, so the structure, the sporangium that contains the megaspore is called megasporangium. Otherwise, the megasporangium is much smaller in size as compared to the microsporangium. So during the formation of the mega and the microsporangium, the Anabaena azollae, the cyanobacteria that is present in the leaf of the in the upper lobe of the leaf, they get incorporated in both the sporangium, so that in later stages, that is during the formation of gametophyte and during the formation of fertilized embryo, this Anabaena azollae, they get incorporated in the next generation. So the basic plan of the sporangium, it is actually a leptosporangiate fern, which means that the initial cell for the formation of sporangia is a single cell. So this type of sporangiate sporangium, the leptosporangiate sporangium, is much smaller in size, and it has its origin from a single cell. And here the sporangium, it is surrounded by parenchymatous sporocarp wall. The sporocarp wall or sporocarp envelope, it is parenchymatous, that is made up of parenchymatous cells, and it is double layered, and this double layered sporocarp envelope protects the sporocarp from mechanical damage or from water. This is how the megasporangium it looks from externally, and it is how, after we remove the outer sporocarp wall, this is how it looks. Note that in Azolla, any megasporangium, it produces initially 8 megasporocyte. This 8 megasporocyte in turn produces 32 megaspore. But of all the 32 megaspore, only one is functional, all other degenerate. So any megasporangium at mature stage will contain just one megaspore. Though it will contain, in the intermediate stage, it might contain 8 megasporocyte to 32 megaspore. So this megaspore, at initially it will contain 8 megasporocyte, which will divide to produce 32 megaspore, and ultimately of all these 32 megaspore, what will happen? Only a single megaspore will be left. So this is the megaspore, this one, and surrounding it is the tapetal cell. On the megaspore, it is surrounded by tapetal cell mass, which can degenerate to form the plasmodium, and also it can form the massula, the elongated structures that surround the egg. They are called the massula. Here we see these are the fibrous part, and they are mainly protective in function so far as the megasporocyte is concerned. And this is the, here in this diagram we see that this region, they dehisce to expose the megaspore. This region here we are seeing that already there is a bit of dehiscence, that is D region. So this dehiscence, the megaspore is exposed. This is the transverse section of the male sporocarp. Here A, this one is the male sporocarp, and we see that inside the male sporocarp there are many sporangia. So actually 8 to 130 number of sporangia can be present depending upon the type of species of Azolla. And within each of these sporangia, these structures, they contain many microspore. So the method of development of the microspore is similar to that of the megaspore, but in case of microspore, all the microspore are functional, that is 32 functional spores will be produced, and that is in contrast to the megaspore, where only one is functional and rest degenerate. And the male spore are much smaller in size to that of the megaspore or the female spore, and both the male and the female spore, when they are mature enough, the microsporangial wall and the megasporangial wall, they dehisce, and the mass of microspore is liberated. Mass of microspore is liberated together. They are entangled within massula, a structure called massula. They are entangled and they come out as a mass of not singly. And this actually maximizes the chance of fertilization. So as in Azolla, in order to maximize the chance of fertilization, the aggregate of the spore comes out. And this is the diagram where we see that we can see both the this enlarged structure, this large spherical structure is the microspore. It is the microspore, and at the basal region of the microspore, we see that the megasporocarp. This is the microsporocarp, and at the basal region of the microsporocarp is the megasporocarp. So both the sporocarp are closely placed, and also when the spore mass comes out, they occur en masse. So this is basically to maximize the chance of fertilization. So this is the structure. Here we are seeing, this is the structure I have been talking about, where this one is the spore mass, the microspore mass here, and they are entangled within this structure. These are the massula like structure here, and we see that the tip of the massula, it is a hooked structure, and that is called the glochidia. So this entire massula structure, it is plasmodial in origin. The tapetum, tapetal mass that surround the spore mass, later it is get converted into this massula like structure. And this glochidia, that is the hooked structure, they help in attachment to the male to the female sporocarp. This glochidia, this hooked structure, they help in attaching to the female sporocarp and maximize the chance of fertilization. So this entire structure, and here we see that as the male spore is formed, the blue green algae, it is also getting incorporated in the sporocarp here, and also here also we see that they are getting incorporated. So basically, when the microsporangia, the sporocarp wall, they disintegrate, and also the sporangium, they also disintegrate, the sporangial wall. Both these two walls disintegrate. This entire mass of microspore along entangled in the massula, it comes out and gets attached or lodged to the surface of the female sporocarp or the megaspore sporocarp inside the spore. That is, the male gametophyte is produced inside the microspore, and the female gamete inside the female, that is the megaspore, and that is, they are produced in a very secure environment. So the megaspore, it produces the male gamete of the female gametophyte, and which contains the female reproductive organ, that is the archegonia. And each megaspore will produce a gametophyte that will contain more than 2 archegonia. And this archegonia, each of this archegonia will contain an egg. And the microspore also, it produces the male gametophyte, that is the antheridia, which produces sperm. And the number of sperm produced by a single antheridia is usually 8. So this archegonia and antheridia are the ultimate reproductive organ, and they bear their gamete, that is the egg and the sperm. So this is the plant of Azolla, the diagram of a plant of Azolla, and a close-up of Azolla, where we see that the Azolla have a bilobed leaf. So this one is the cyanobacterial portion, and this one is the reproductive portion. So in the reproductive portion, we are seeing that this is the male sporocarp, and this is the female sporocarp, where the male and the female spore are produced, and the spore inside the spore are this is the spore mass, and each of the spore mass contain many spores, and the spores, this microspores or the male spore, each spore produces archegonia, that produces each archegonia produces 8 number of sperms. And this one, the female sporocarp, it produces the female spore, that is the megaspore, the female spore. Each of this female spore produces archegonia, and many archegonia. Each of this archegonia will contain one egg. This is the diagram of male gametophyte, where we see that so basically these are the hormones, this is how the filaments egg cell, if allowed to fertilize, the cyanobacteria Anabina azole, that is getting incorporated in the male gametophyte. And this is the antheridia. This is the antheridia in formation that produces the sperms. So when any microspore, it is ready to germinate, what happens is that the exine dehisces and the intine, along with the content, forms a germ tube, and by repeated division of the germ tube, by actually first the first division, a very large cell is produced and a very small lenticular cell is produced. So this small lenticular cell divides and redivides and ultimately form this antheridia, which produces the sperm. And this basal cell, this large basal cell, it is mainly of nutritive in function, and it forms forms the nutritive matter, and no further process in the formation of spermatocyte, but the small lenticular cell, this they ultimately divide redivide to produce the antheridium, and in the antheridium we can see ultimately 8 spermatocytes are produced. The antheridium of this Azolla, it has a single jacket layer that is protective in function, and inside the jacket layer are the antheridoid mother cell, and this mother cell, by repeated division, by reduction, this 8 number of spermatocytes are produced, and these 8 spermatocytes, they are flagellated. So it is a swimming type of reproductive germ cell. It also, when it is mature enough, there is dehiscence of the outer wall, and the inner content, it forms a very large basal cell and a terminal lenticular, small lenticular cell by division. And this lenticular cell here also, the lenticular cell take part in formation of the archegonium, which harbors the egg cell, and all these things, they are happening inside the megasporangium, that is harbored inside the megasporocarp. So here the development of the gametophyte, the female gametophyte, occurs inside the female sporocarp. So this type of in the developmental stages of Azolla, it is very much similar to the another aquatic fern, that is Salvinia. And actually I have made a detailed video on reproduction of Salvinia. You can just go to that video just to have a comparative study of Azolla and Salvinia. I've put a link of the Salvinia video on the description box, so you can go and also have a comparative study. So basically this megaspore, this is how the small lenticular cell, the terminal lenticular cell, and the large basal cell, and this is how the female gametophyte is produced. And as the female gametophyte is getting produced, the upper protective layer, it is getting removed. So this is how pretty much the female gametophyte is produced, which harbors the archegonium that contain the egg. So this is how the sperm looks, and when the sperm is mature, and they are the germination of the microspore and formation of sperm inside the archegonium, they happen while still inside the massula like structure. That is, all the microspore, they germinate and produce sperm, and all those are staying together inside in the mass called the massula. And so we see that this is basically there will be maximum amount of sperm for the egg, and just for increasing the reproductive potential, this type of process is done. And this is the archegonium, where the neck cell, they have already dissolved. Ventral canal cell, they are in a process of dissolving. They will also dissolve and create a very mucilaginous tunnel that attract the sperm. So the sperm will enter this neck region, ventral region, and ultimately fertilize with the egg and produce a zygote. Now, as because the during the formation of archegonium as well as the formation of antheridium, the inside the spore. A large amount of Anabina azole hormogonia has been introduced. So during the formation of the zygote and the embryo development, this Anabina azole also gets incorporated inside the developing embryo. And this is the picture of the upper lobe of the plant, where this Anabina azole contained inside the gas vacuole of the upper lobe, and this Anabina also gets transported to the embryo region. So, after fertilization, embryo for zygote is formed, and zygote by repeated division forms the embryo. And in the division, the first division is always transverse, followed by a vertical division, and to produce a quadrant. And in the quadrant, that is the lower quadrant, foot and root are produced from the lower quadrant, and from the upper quadrant, the stem and leaf are produced from the upper quadrant of the embryo. And the foot, though it is cylindrical, it does not continue growth for long, and its growth of the foot region, it remains very limited period of time. And after the foot growth has stopped, we see the growth of other parts, that is the root, the stem, the leaf. Though the growth of the growth rate for the root is very slow as comparison to the root and the stem region. So the ultimately embryo divides to produce this type of structure. So if there is a recapitulation, we see that this is the mega spore, that is the female spore. This is the microspore, that is the male spore, and male spore dehisces, the sporangium dehisces. And this is the massula, which contain the spore and entangled in this type of glochidia. This is the glochidia. So this entire massula containing the spore, and all the spore, they germinate, produce the gametophyte, and ultimately produce the sperm while still inside the massula. And so this is the antheridium. This is the antheridium which produces the sperm. This is the archegonium, and ultimately there is a fertilization of the sperm and the egg. So and ultimately the life cycle where we see that sporophytes are produced. Adult plant which mature enough, and it produces the the female sporocarp or megasporocarp and the microsporocarp. Here this is the microsporocarp here, and this one is the megasporocarp, where I'm writing it in capital letter, megasporocarp. For the female sporocarp, I'm writing in capital letter. So the megasporocarp contains this megaspore, which is so the reproduction division occurs in the spore formation period. So the megaspore and the microspore. So microspore, that is it is the male spore, and this one is the female spore. So ultimately the spore, they germinate to produce the archegonium, where this one is the egg cell, and the antheridium, it produces the flagellated sperm. So ultimately the sperm fertilize the egg, and ultimately zygote, that is a diploid structure, is formed. And so zygote is the first diploid structure. So up to this one is the haploid stage here in this region, and the rest one is the diploid stage. So the zygote It forms the embryo by repeated division. Ultimately, this is the embryo which produces the mature sporophyte. So we see that right from the zygote to the sporocarp, that is here, to the sporocarp region, this is the diploid stage. And once the spore is produced, the haploid generation begins. Haploid generation begins with the spore and ends with the egg and the sperm or the anthrozoid, and rest are all diploid stages. So this is the life cycle. Life cycle of Azolla. Hope you like this video, and if you like this video, please do share it with your friends, and please do subscribe, because your subscription, it encourages me. And those who have subscribed, thank you for the subscription. And if you have any want me to make any videos, do write in the comment section. And those who have already commented, I am making all your videos one by one, so please be patient. And thank you again for watching this video.